Assalamu alaikum wa rahmatullahi wa barakatuh. We are going to proceed with activity 2 of chapter 3, the gene structure and information unit, part 2. At the end of this video, you will be able to note that the nucleotides in a gene are considered 3 per 3, called codons, and a codon is a triplet of nucleotides. Recognize that each codon codes for an amino acid. Note that some codons code for the same amino acid and others don't code for any amino acids. Deduce that a change in the codons of a gene may lead to the formation of an abnormal protein. As we studied previously, a gene is a fragment of DNA characterized by a determined sequence of nucleotides that codes for a specific protein. A protein is an association of amino acids. But how does a gene that is a sequence of a nucleotide code for a specific sequence of amino acids? In fact, we have only 4 types of nucleotides: A, T, C, and G. And we have 20 types of amino acids. So we wonder about the number of nucleotides in the DNA sequence that code for one amino acid. Then we can pose the following problem. How many nucleotides in the DNA sequence code for an amino acid in the protein molecule? To answer this problem, we can pose different hypotheses. The first hypothesis is that each nucleotide codes for one amino acid only, which means that the first nucleotide, which constitutes of phosphoric acid, deoxyribose sugar, and nitrogenous base A, codes for a given amino acid. Similarly, for the second nucleotide, which contains nitrogenous base T, codes for a different amino acid. A third nucleotide codes for a third amino acid, and the fourth nucleotide codes for a fourth amino acid. But in this case, we get only 4 types of amino acids, coded by the 4 different types of nucleotides. That is, according to this hypothesis, if we apply this rule, 4 to power n, n corresponding to the number of nucleotides involved to get an amino acid, we get then only 4 types of amino acids. Okay, but in reality, we have 20 types of amino acids that exist. So this hypothesis is rejected. As for the second hypothesis, every 2 successive nucleotides belonging to the same strand of DNA code for one amino acid, like A and T code for one amino acid, A, C code for another amino acid, and so on. So according to this hypothesis, if we apply the rule 4 to power 2, this gives 16. That are 16 types of amino acids only. And this is not true, because in reality there are 20 types of amino acids. So this hypothesis is also rejected. As for the third hypothesis, it says every 3 successive nucleotides, which is triplet of nucleotides belonging to the same strand of DNA, code for one amino acid, like A, T, C correspond to one amino acid, A, T, G code for another amino acid, and so on. According to this hypothesis, if we apply the formula 4 to power 3 equals 64, then this means that there should be 64 types of amino acids. But in reality, we have only 20 types of amino acids that exist. So how can we explain such a result? First, let's confirm the number of nucleotides coding for one amino acid. Here we are given this example, an amino acid sequence of the B chain of the normal insulin molecule and its corresponding DNA molecule. As we notice, every amino acid is encoded by 3 nucleotides. These 3 successive nucleotides are described as a triplet or a codon. So every 3 nucleotides are said to be a triplet or a codon, and they code for one amino acid. Here you can see the genetic code table It is a table that includes all the possible codons and the amino acids that are coded by different codons. To easily find the codon you are looking for, you should look for the first letter horizontally, the second letter vertically, and the last letter horizontally. Let's take an example. If we are looking, let's say, for AGA, horizontally concerning the row that contains letter A, and then vertically G, we have to look in this cell here, okay, and the last one also horizontally, so AGA. This is it. And for now, we forget about letter U. We will discuss it in the next activity. If you go back to the problem that was posed previously concerning the presence of 64 codons and the fact that we have only 20 amino acids, this can be solved by the fact that more than one codon encodes for the same amino acid, but each codon cannot encode for more than one amino acid. So we get into this conclusion here. The 64 codons are classified into 61 coding for amino acids, including the start amino acid, which is called methionine, and we have 3 stop codons which encode for nothing. If we go back to the table, we can see that these are the stop codons, UAA, UAG, and UGA. And this is the start codon, AUG, which encodes for methionine. In normal cases, after the protein is produced, it performs its function. However, in some cases, the produced protein would be malfunctional or inactive, or its function has been modified. What could be the reason behind that? In normal cases, normal proteins are produced because the DNA has a normal sequence. Then these normal proteins would perform normal functions. However, in other cases, if the DNA sequence is abnormal, then the produced protein would be abnormal and its function would be modified. Let's take an example here. Here we have 2 sequences that belong to a normal insulin molecule and, on the other hand, an abnormal insulin molecule. If we compare both sequences, we notice that the difference between both molecules is at the level of this amino acid. Instead of histidine, we have aspartic acid. And the reason behind that is that one of the nucleotides has been changed. Instead of C, we get G. The change in the DNA sequence leads to a change in the amino acid sequence, and thus the whole protein molecule would have a different, modified, abnormal function. So, if one nucleotide differs, we may get a different amino acid, and in this case, the produced protein would be inactive. To sum up, a gene is a fragment of DNA coding for a specific protein. A gene is made up of a series of nucleotides considered as codons, where the codon is a triplet of nucleotides. Each codon codes for an amino acid. Some codons code for the same amino acid, and others don't code for any amino acid. A change in the codons of a gene may lead to the formation of an abnormal protein. Thanks for listening.